for the next 6 minutes, we have 3 presenters here for the first Ignite talk. It was fantastic how many groups actually approached me about that. As I said, first come, first serve. So I gave the 3 Ignite talks to the 3 people for the 3 groups who approached me first. So take the stage, please. The mics. Just one? Okay.
Hi, I'm Julia.
I'm Shoshana.
And I'm Marianne. And today we're going to be talking about mutation and selection through the lens of HIV. So what is HIV? Human immunodeficiency virus, commonly referred to as HIV, attacks the helper T cells in the immune system. These T cells play a major role in cell-mediated immunity by identifying and attacking pathogens. So without them, people struggle to fight off infection. HIV is transmitted through blood-to-blood and sexual contact, and it is a retrovirus, meaning that its genes are made up of ribonucleotides rather than deoxyribonucleotides. Currently, 33.4 million people are living with HIV, and over 25 million have died of its causes since 1981. This disease is most prevalent in South Africa, Nigeria, and India, but there are also over 1 million cases in America. So how does HIV work? HIV becomes attached to the white blood cell when the GP120 protein binds with the CCR4 and CCR5 glycoproteins on the cell surface of the helper T cell. The viral core then enters, and the membrane of the GP120 protein fuses with the membrane of the helper T cell. Then, the virus uses reverse transcriptase to copy its RNA into DNA. The new DNA is now able to create mRNA using its own enzymes, which is then translated into viral proteins. These proteins then pinch off from the cell membrane and attack helper T cells, depleting the immune system and making it much more prone to infection.
So you're probably familiar with HIV, but did you know there's a mutation that can give you immunity to it? The mutation is found on the CCR5 gene located on the 3rd chromosome. This mutation is known as CCR5 delta 32 mutation. It prevents the helper T cells from developing the CCR5 receptor, as we just saw, before the HIV viruses that used to invade cells. In order to have full immunity to HIV, you must inherit this mutated gene from both parents. If you inherit it only from one parent, HIV symptoms are only delayed. Now that we're talking about mutations, let's have a brief review of what we learned in this class. As we know, mutations are the mechanisms of natural selection, and they affect proteins via the central dogma. Mutations exist in DNA, which is transcribed into mRNA, which ribosomes use to create proteins, which function as enzymes, receptors, and many essential molecules. In class, we specifically covered point mutations, which is the addition, deletion, or substitution of a single base pair. Types of point mutations that we learned about include nonsense mutations, missense, frame shifts, and silent mutations. Another type of mutation are chromosomal mutations. These include, for example, deletions or insertions or translocations of multiple base pairs. But let's get back to HIV. The mutation that results in HIV resistance is a 32 base pair deletion in the CCR5 gene. Mutations sometimes cause losses of function or gains of function. This can be beneficial or harmful depending on the situation. In the case of HIV, it is a loss of function mutation because we lose the receptor that HIV uses. However, it is beneficial because of the immunity it gives.
All right, so why is there a picture of the smallpox virus up on the screen when we've been talking about the HIV virus this whole time? Well, the smallpox virus is relevant because scientists have found that the same mutation that causes resistance against smallpox also causes resistance against HIV. So about 700 years ago, there were a lot of smallpox outbreaks in Europe, and people carrying the CCR5 mutation were selected for. So today, people of European and Central Asian descent are the people that we see with the CCR5 mutation. Now, this is a form of directional selection, just like we learned in class. So one extreme is favored, and in this case, it's the people with the CCR5 mutation because they're more likely to survive. We also learned about stabilizing selection, which is where the middle is favored. So a popular example of this is birth weight, because children of average birth weight are going to be favored for, as opposed to people with really heavy birth weight or a lower birth weight. There's also disruptive selection, and a classical example of this are the finches, like we learned in class. So finches with big beaks are going to be selected for because they can eat the bigger seeds, while finches with smaller beaks are going to be selected for because they can get to the smaller seeds. But then finches with medium beaks are good for nothing and can't get the food, so they'll die off. Okay, so the Berlin patient, who is this guy? So this man was infected with HIV at early years, and he went through the conventional treatments with the concoction of medications, and this only lessened the symptoms, but it never really got rid of the HIV virus, and this is what people are taking today. But unfortunately, later on in life, he was diagnosed with leukemia. So he went through stem cell treatment, and he didn't just use any stem cells. His doctor thought it would be a good idea to try using stem cells from a donor who had the CCR5 mutation. And so after going through all these treatments, to everyone's surprise, the Berlin patient was cured of both leukemia and HIV. This guy's the first guy to be completely cured of the HIV virus. And this is an exciting avenue for the treatment of HIV and has shed a lot of light on the possibility of finding a cure for the disease. Thank you. Do you guys have any questions for us?
Any questions in the audience? They're in a stage of shock. Great, thank you very much.